सुना दे रहा है, क्लियर नहीं है। अभी क्लियर है? आप थोड़ा बाहर हेलो, आपको बोलिए।
हेलो जी सर, हाँ क्लियर सर, हाँ क्लियर है, ऑलमोस्ट क्लियर है।
सुना दे रहा है? क्लियर है?
Yes, sir, it is clear, sir.
ओके गुड।
Pratap, sir.
यस,
Sir, with your permission, can we start the event?
श्योर श्योर श्योर।
Okay, sir. Sure. Namaskar and good morning to everyone. It's my privilege to welcome our distinguished guests and esteemed faculty members and our dear colleagues. Being a successful session for the last 3 consecutive days, today we have stepped into the day 4 of our EFDP session, which is entitled as Empowering Technological Advances in Pharmaceutical Education and Research, thoroughly been organized by Bharat Pharmaceutical Technology. On behalf of the organizing committee of EFDP and Bharat Pharmaceutical Technology, I, Arnab Majumdar, Assistant Professor of Bharat Pharmaceutical Technology, heartily welcome you all. Today, we are fortunate to have with us an incredible and insightful speaker, Dr. Pratap Chandra Acharya. To introduce our speaker, I would like to request Mr. Shurujit Shah, Assistant Professor, Bharat Pharmaceutical Technology, to kindly grace our speaker.
Uh, Thank you, Arnav sir. Pleased for giving me this opportunity. A very good morning to everyone. I would like to tribute my cordial thanks to our respected eminent speaker, Dr. Pratap Chandra Acharya sir, for becoming the integral part of our EFDP program. Respected participants, today we are having a wonderful academician and a very dedicated researcher belongs to the holy land of Odisha. Dr. Pratap Chandra Acharya sir at present is working as an assistant professor in the Department of Pharmacy, Tripura University. Sir pursued B.Pharm from Behrampur University, Odisha, then M.Pharm IIT BHU Varanasi, then sir completed his PhD from Punjab University, Chandigarh in 2013. Sir has a keen research interest in heterosteroid synthesis and steroid modifications toward anti-cancer drug discovery, as well as synthesis of lipid drug bioconjugates and heterocyclic small molecules to target colon cancer. Sir has also research interests in synthesis of glycolipids for nanodelivery of anti-cancer drugs, as well as method development for the analysis of pharmaceuticals, drugs, metabolites, impurities, and degradants. Sir has completed several post-PhD research projects like impacts of air pollution on respiratory health from the University of Edinburgh, Scotland, UK, funded by Commonwealth Scholarship Commission, UK, as well as screening of spiroxyndole derivatives as cancer chemotherapeutic agents by targeting G quadruplex interaction from Research Institute for the Medicine, Faculty of Pharmacy, University of Lisbon, Portugal, funded by EMBO, Heidelberg, Germany, as well as sir completed post-PhD research work in role of NLPL PRC complex on MEPS regulation in E. coli from Center of Cellular and Molecular Biology, Hyderabad, funded by INSA, IASC, and NASI, India. Sir has achieved n number of awards. Commonwealth Professional Fellowship 2023 by Commonwealth Scholarship Commission UK. Then Faculty Awards, Certificate of Honor and Medal by Tripura University in 2019. Then European Molecular Biology Organization Short-Term Fellowship 2018 Award. Then Faculty Award and Certificate of Honor by Tripura University in 2017. Then DST Young Scientist International Travel Grant Award by Department of Science and Technology, Government of India in 2017. Then Science Academics Summer Research Fellowship in 2017, jointly awarded by Indian Academy of Science, Bengaluru, Indian National Science Academy, New Delhi, and the National Academy of Science, Allahabad, India. Best Paper in Medicinal Chemistry Award and Best Oral Presentation Award at 63rd Indian Pharmaceutical Congress, Bengaluru, India in 2011. Research Project Presentation Award in Mansodon 4, Mumbai. Then UGC Research Fellowship for Meritorious Student in Science in 2009. Then Gate Fellowship in 2006. Sir successfully completed several sponsored research projects funded by UGC New Delhi, as well as CSIR New Delhi, DBT New Delhi, ICMR, AICT from several funding agencies. Recently, Sir is running a wonderful project entire in the Northeast, that is establish a drug metabolomics laboratory for Northeastern states, which is funded by DBT New Delhi, around 476.26888 lakh. That is the sanctioned budget of this project. So, Sir successfully filed 2 patents. Sir has published near about 22 research papers in international research papers with high impactful journals. Some of them are from Drug Design and Discovery, Journal of Molecular Structure, like that. Sir has edited book in Medicinal Chemistry of Chemotherapeutic Agent, a comprehensive resource of anti-infective and anti-cancer drugs, first edition, that is published in 2023. Sir has published a number of papers, book chapters, as well as Sir has presented and chaired numerous conferences and proceedings. Sir already transferred technology to the industry, and Sir has a huge collaborations with different universities like University of Tennessee Health Science Center, Memphis, TN, United States, as well as University of Lisbon, Portugal. Then the Department of Molecular Biology and Bioinformatics in Tripura University. Then Bombay College of Pharmacy and Department of Pharmaceutical Science, ICT Mumbai. Then IIT with IIT BHU. So numbers of collaborations are also having, Sir, with different universities, reputed universities. As well as, Sir, supervising number of PhD scholars, already supervised PhD scholars, and more than 21 MPharm students already supervised. Sir is a member of Research Advisory Committee of PhD thesis at Tripura University, as well as SVKM NIMS University. And Sir is appointed as a reviewer of several leading journals such as Therapeutic Delivery, Journal of Pharmaceuticals and Biomedical Analysis, as well as Journal of Chromatography, Human Cell, Scientific Reports, Current Pharmaceutical Analysis, like that. Sir, having the administrative responsibilities also, sir was HOD in Tripura University of the Pharmacy Department in 2019 to 2023, in charge of Research and Innovative Cell, Tripura University, then convener and member of Board of Postgraduate Studies, Department of Pharmacy in Tripura University, as well as member Board of Faculty, Faculty Studies, BFS, Faculty of Science, Tripura University, and playing a very important role in the member of various committees of the university related to PhD studies, academic and administrative matters. It is a very difficult to brief the contributions of our respected speaker in this very short span of time. Sir, we are really excited and delighted to learn from your today's presentation. Thank you very much, sir, with for sharing your experiences as well as sharing our EFDP program, contributing in our EFDP program. Thank you very much. We are very much delighted, sir. Thank you. Sir, stage is yours. You can present, sir. Please proceed, sir.
Sir, am I audible now?
Sir.
Am I audible?
Yes, sir, you're audible.
Thank you, Arnav and Surajit sir, for a elaborative introduction to the audience. And I am also thankful to Dr. Subhasis Devna for inviting me to this August gathering. And so with this, I'll start my presentation. So let me know if it is visible to you. Is it visible?
Yes, sir, it is visible, sir.
I hope the presentation is in the full screen mode and it is visible
No, sir,
to everyone.
actually, I think you need to go to the slide share mode. I think then it will be visible.
1 second. I'll do it again. Now I've set the screen. Is it visible in the full screen mode?
Sir, you just now it is visible. Yes, sir, it is visible now, sir.
Okay. So I may not do the video because I need to stand and there are audience in front of me, so I have to address them also. Okay. So today, my topic of discussion is strategy to combat cancer, bombing the chemotherapy at the doorstep of the malignant cell. Let us understand what is it. There is a very old saying that cancer has no answer. Have you heard of it? You must have heard of it. Cancer has no answer. That means there is no treatment for cancer. Right? So how do we tackle that? How do we treat cancer? How do we combat cancer? So there is one technology or one tactic that can be adopted is bomb the chemotherapy at the doorstep. That means you have to take the chemotherapy and put it at the doorstep. Doorstep मतलब इसके द्वार में जाके इसको doorstep में जाके इसको delivery करना. So you can just put at the doorstep of the cancer cell and the cancer cell will die. So let us understand step by step how it goes. Okay. Now, how do we define cancer? So a British oncologist, R.A. Willis, gave the definition, the shortest definition of cancer. It is a disease that is untreatable or that progresses even after the stimuli that has started the disease has been stopped. So it is also known as neoplasia, which means new growth or a tumor. And you know that this is of two types, benign and malignant. So benign means it is benevolent or innocent. It will not harm anything. But malignant means malevolent or evil. When we talk about evil, evil means it is like a demon. So what is a demon? A Ravan. So you can take the example from the mythology, Ramayana mythology, that Ravan was a evil. So cancer is nothing short of a Ravan. Okay, so cancer is the leading cause of death. Around 30% of death worldwide happens due to cancer. And whatever chemotherapeutic agents we have in our hand is not sufficient to treat cancer, which means even though we have treatment options, we have drug molecules, it is not sufficient for the treatment of cancer. And especially when there is metastatic spreading. What is metastatic spreading? Cancer when starts from one organ and spreads to other organ is called metastasis. And once metastasis cancer happens, the patient is destined to die, right? And there is another concept called drug resistance. Most of the cancer drugs are now resistant to the chemotherapeutic agents. So these are the demerits of having cancer treatment. So let us understand how the cancer timeline has started. Is it visible? So cancer is not a
Yes,
new
sir, it is
disease.
visible.
Is it visible?
Yes, sir, it is visible.
Okay. So cancer is not a new disease. It has been identified 80 million years ago in a dinosaur bone fossil. So when the scientists examined the bone fossil of a dinosaur, they found that the dinosaur died because of cancer and it had a bone marrow cancer. 3000 BC, 3000 before Christ, you can see those two surgeons are performing some kind of experiment on a mummy. It is an Egyptian mummy found in the Egypt, and they found that this mummy, has died because of cancer. Not a new disease. So the oldest description that describes the appearance of cancer is papyrus, the Smith papyrus, Edwin Smith papyrus. It is a papyrus means paper. So this was a paper that was found in the Egypt, which has written description of cancer. So we cannot understand what is written here, but those person who can understand the Egyptian language can see that the cancer has been described, and it has also described that there is no treatment for cancer. How many years ago? 3000 years ago, 3000 plus 2000, 5000 years ago, it was written that cancer has no treatment. Now, we must be knowing this person, Hippocrates, father of medicine. 4000 before Christ, he was the first person to describe cancer as carcinoma. Carcinoma, right? And he is the person who coined the term cancer. So he also suggested that there is no treatment for cancer, right? So we saw that the papyrus, Edwin papyrus described that cancer has no treatment. The father of medicine, Hippocrates, told that cancer has no treatment. Then came a scenario in 1846, Morton. Morton was performing so he discovered anesthesia, ether. Have you seen diethyl ether in your laboratory? You must have seen. So diethyl ether is a very volatile solvent. When you inhale diethyl ether, you can get anesthesia. It can make your body go completely into coma, completely into anesthesia. So ether was the best anesthesia, general anesthesia discovered in that era. But now we have more modern and more profound anesthesia, so we have discontinued the use of ether. So what he is doing, just like a Bollywood movie, he is putting some ether into the towel or the handkerchief and putting on the nose of the person. He is getting anesthetized. बेहोश हो गया. You have seen it in Bollywood movies, right? So using that ether as anesthesia, he is doing a dental extraction. उसका दांत निकाल रहा है. He's a dentist. And using that ether, William Halsted first time in 1880s, he performed a mastectomy, breast cancer का surgery कर रहे हैं. So you can see there are doctors surrounding a cancer patient and doing surgery. How? By doing the anesthesia using ether. Now, this revolutionized the cancer therapy, that is surgical removal of cancer or cancer tissue. So in 1903, yes. So Marie Curie discovered radium. You must have heard about Marie Curie. So he discovered radium, and radium was used as a radiation therapy for cancer. To treat cancer, you can use radium as a radiation therapy. It was in 1903. And in 1950, 1945, after the Second World War, Gilman and Goodman, two pharmacologists, you must have heard their names, Goodman, Gilman, book of pharmacology. So they were the first person to introduce nitrogen mustard as cancer chemotherapy agents. Nitrogen mustard, the carbustine, lobustine, all those nitrogen mustards were used for the treatment of cancer, for killing the cancer cells. So it was in 1950. Now, how much we have progressed from 1950 till date, till 2025? It has been 75, more than 75 years. And how many drugs we have? We have almost 780 FDA-approved anti-cancer drugs. That means we have medicines for cancer, but what we do not have is that the treatment for cancer. Although we have 700 medicine nowadays, in the present day, we cannot treat cancer using any of the medicament that is available in the market or that is approved by FDA. So this is a screenshot that I have taken from the NCI website. So these are the list of medicines and the combination therapy that are available. This is list of A. So you can just click on A, B, C, D, E, F, G, Z, and you can find all the name of the drugs that have been approved by FDA, but none of them are useful for treating a cancer patient. Now, what we have in our hand? We have three different categories of medicines, drug molecule, available for the treatment of cancer. One is tumor specific or tumor selective agent. So what does that mean, tumor selective? Because that means it is very selective to the tumor. For example, rituximab, it is a monoclonal antibody. So it is very particular for Hodgkin's, non-Hodgkin's lymphoma, right? So even though you have your body cells rapidly growing tissue cells, the rituximab will go and attack only a non-Hodgkin's lymphoma. So this is called tumor specific. They are very specific to a type of cancer. And you have tissue selective agent. So tissue selective means these agents select the tissue. For example, aromatase inhibitor or 5-alpha reductase inhibitor. Aromatase inhibitors are the drugs that only inhibit conversion of testosterone into and or androstenedione into the estradiol. So it happens only in the breast tissue, breast cancer case. 5-alpha reductase inhibitor. So 5-alpha reductase inhibitor is an enzyme which is used for the conversion of testosterone to 5-alpha hydroxytestosterone, the active metabolite of testosterone. And this inhibitor will act only in the benign prostate hyperplasia or the prostate cancer, only in male tissue. So they are very specific to tissues. Then we have cytotoxic agent. Cytotoxic agent means it will kill the cell, irrespective of it is whether cancer cell or your normal body cell. So there are numerous fastest growing tissues in our body. For example, hair, it grows every day. So that is why when you give a cancer treatment to a person, the person has to get his or hair saved first, because it will make you bald, right? Skin, our skins are very much rapidly growing. So if someone is taking the anti-cancer drugs, then what will happen? Your skin will get eroded, you will get blisters, you will get red patches on your body. So that is why you need to have your other medications like steroids, ready in your hand, right? But cytotoxic agents are the most profoundly available anti-cancer drugs that have been approved by FDA, right? So what will happen? So these are very much cytotoxic. So when we take these medicines, the quality of the life of a person will worsen. What does that mean? You will kill cancer. You may be able to kill the cancer cell, but at the simultaneously, you will also be able to kill the person itself. वो बंदा ही मर जाएगा तो फिर कैंसर को ट्रीट करके क्या फायदा, right? So these are the demerits of the medications that are available for the treatment of cancer. Why it is difficult? क्यों कैंसर को ट्रीट करना डिफिकल्ट होता है और why it is very difficult to target cancer cell? I'll give an example. This is the first category of drug that we saw is the tissue selective. Sorry, one second. So that is the tumor specific drugs. So this is a kinase enzyme family. Kinase enzyme is involved in the development of cancer in various ways. So if you inhibit kinase enzyme, the cancer cells will be killed. Now the question is how many kinase enzymes are present in our body? You can just imagine this is the family tree. This is a family tree. So in their family, there will be father, there will be mother, there will be son, there will be daughter, there will be grandson, granddaughters, etc., etc. So these are only families, Saha family, Acharya family, and all family. In that family, there will be other members also. So just imagine how many different type of kinase enzyme are there in our body. So suppose the scientists want to develop a drug molecule, which kinase enzyme they have to inhibit? They have no clue. So even if you enzyme one kinase enzyme, there is possibility that all other family will also be destroyed. So what is treatment? You are just bombing it. So chemotherapy, just imagine that chemotherapy is a bomb. Now you want to bomb the chemotherapy. Suppose Israel is now bombing the Gaza Strip. So what is happening? All the Gaza Strip is getting erased out. So what is the target? That target is the Hamas strip. But what they are doing, they're erasing an entire country. So you cannot bomb everywhere. So, but our strategy is like that: we are bombing everywhere in our bodies so that the cancer will also get destroyed. But in that process, cancer will also get destroyed. But the worst part is that person will also get destroyed. So that is why the topic is we want to bomb at the doorstep of the cancer, at the doorstep of the terrorist, or at the doorstep of the person or the causative agent. So that is why if you target cancer, it is very difficult because if you put a chemotherapy, just imagine chemotherapy is a bomb. You want to kill this cell line or this kinase enzyme, the blue dotted kinase enzyme. So if you put this chemotherapeutic agent, it will not only kill the blue dotted kinase enzyme, it will also kill the green, red, and all other dotted kinase enzymes, which is not advisable, right? We want a mechanism which will go and only attack only one at one family or one point at one doorstep. This is the topic of the discussion. So this was about the enzymes. Similarly, there are cellular mechanisms. In a cell, cancer cell adopt different mechanisms. So they can inhibit mitochondrial integrity. They can bypass the autophagic relationship. They can bypass the cell checkpoints. They can do numerous kind of bypassing mechanism so that the cell will grow. It is like you are killing rubber. So you have heard about the Ramayana mythology where rubber has 10 heads, and if you shoot one head, or the Ram used to shoot one head, so another 10 heads used to come up. It is like that. So if you block at one point, if you block the metabolic pathway, it can have a ROS pathway to bypass that, or it can get some other pathways in the cell to grow itself, right? So it is very difficult to kill the cancer cells. So these are summary of the things that a cancer do in a oncogene to downregulate itself so that the cell continues to grow. So even if you check at one point, there are 10 other points where the cancer can pick and can grow. Suppose in a war zone, a king is inside his castle, So how the enemy has surrounded the castle from the outside, and the enemy want to go into the castle to kill the king. What they will do, the enemy will first block all the doors. So if you block all the doors, foods from outside, food and water from outside cannot enter into the cell. And whatever supply is available in that castle, the king or queen will utilize that and survive till 1 month or 2 months. After that, supplies will be less. Supplies cannot come from the outside. The castle will fall down. Right. This was the tactics used by the enemies during the ancient times to kill kings, kings that they have been reserved in the castle. To just block their doors, block their supplies. So this is how we can block the cancer to survive. The cancer cell, so if you want to block this one, the cancer can take the food from this door, right? So there are numerous doors available for the cancer. Even if you block all the doors, they can create another door and bring the supply inside, right? So this is why targeting cancer is very difficult. Now, after discussing all those things, what we understand that there is a problem to treat cancer, and the major problem is there are too many things to be targeted at one time. You have to block many doors at a time to kill the cancer cell, right? Now, it is not possible to block all the door or many doors at one time to kill the cancer cell. So what we have seen that initially the tumor may shrink, but afterward again the cancer will revive. Now, secondary cancer कहते हैं इसको. It is called secondary cancer. Many person who undergoes chemotherapy, after few years they will develop another cancer, especially leukemia. Suppose a person is getting treated for breast cancer or prostate cancer. After 5 years, you will get cured. But after 5 years or 10 years, you will get another cancer, blood cancer. Right? So this has been seen with the cancer chemotherapy. If you block one door, cancer is going to open another door for itself. So what is the solution? So if this is the problem, what is the solution? So the solution is that you eradicate the oncogenes. What is oncogene? A gene that initiates the cancer cell to proliferate. So there is a gene which the oncogenes are cancer-causing genes. There are genes. So if you can eradicate, or you can target the oncogenes or proto-oncogene or the same cell that initiate the cells to proliferate, then we can achieve the cancer chemotherapy in a orderly manner. Or you can develop combination therapy. Combination therapy means you can add more than one chemotherapeutic agent. You can add more than one chemotherapeutic agent to destroy the cancer. Now, as a pharmacy student, as a pharmacy professional, if I want to ask you, what is the therapeutic strategy for cancer? If some of your neighbors or someone has cancer, what the oncologist will do first? Will they give medicine? Do you think that they will give medicine at the very beginning? No. The first option to treat cancer is that you give a surgical removal, or you remove the cancer surgically. No, the first line of therapy is surgery. But modern started anesthesia करके, so you need to remove the cancer. So this is the first line of therapy, that you have to surgically remove the cancer. But then there are cancers which cannot be surgically removed. Leukemia, for example, blood cancer. You cannot cut the blood. Lungs cancer, you cannot cut the lungs. Person will die. Gastrointestinal tract cancer, colon cancer, stomach cancer. If you cut stomach or colon, person will die. They are smooth muscles. So what is the second line of therapy? The second line of therapy is radiation therapy. What Dr. Marie Curie developed, the radiation therapy using radium. So just focus the radiation, gamma rays, beta rays. So they will destroy the genes or the cancer cell malignant cells. The cancer may be eradicated. This is the second line. So you have to do surgical removal of the cancer if it is a solid tumor. So once the tumor is removed, then you have to put the gamma radiation or the radiation therapy to remove, even if some cells are surviving. हो सकता है there are few cells which are still surviving even after the surgery. So that you need to kill. So that can be killed using radiation. But if you put radiation in the first line as a first line of therapy, so that can also induce cancer. Radiation can induce cancer. Like Marie Curie died because of cancer because of radiation. He was working on the radium metal. So the radiation from the rhodium metal came into her body and she got cancer, breast cancer, and she died. So radiation cannot be the first line of treatment. It is always the second line of treatment. And what is the third line of treatment? No, chemotherapy is also not third line of cancer treatment. Third line of treatment is neoadjuvant therapy. So what is neoadjuvant therapy or adjuvant therapy? So you have to do the surgery, add the radiation, and put some chemical reagent or some chemotherapeutic agent which will not harm the body itself. If all the three fails, then we go for chemotherapeutic agent or the drug treatment. Because chemotherapeutic agent has its own adverse effect. It can damage your own tissue, faster growing tissue like your hair, alimentary canal, your skin, gonads, testes, ovary. They can degenerate everything, all the fast growing tissues. So what is the strategy? The right strategy. So if you have heard about Raman or if you have studied about Raman, so Ram was first cutting the head of Ravan. So just imagine the cancer cell is nothing but Ravan. It has 10 heads because it can create its own heads. It can create its own door to bring out the supply and survive. It will not die. He has get the boon that it will not die in any normal situation. So when Sri Ram was cutting the heads of Ravan, so if one head falls, then another head will come up. So that means the cancer can adapt itself. If you block one place, it will come up with another mechanism to survive itself. So you cannot cut the heads. So he failed. Now, what is the right strategy? So this was a wrong strategy by Sri Ram to kill the Ravan. So just imagine this is a wrong strategy if we want to hit the cancer cell just by using therapeutic agent. It's a wrong strategy. What is the right strategy? The right strategy is that there is a nucleus, navi कहते हैं इसको. The midpoint of your body is called navel point. Nucleus of the cell, cancer cell, is called the midpoint. So there is Amrit color. So you can take a bow, or this bow is nothing but a chemotherapeutic agent. Just put the chemotherapeutic agent at the navel region or the nucleus of the cancer cell. So that will destroy the Ravan or the cancer cell. If you want to kill the cancer cell, don't put the drugs onto its cell membrane. Cell membrane had all the receptors, so there is where all the chemotherapeutic agent will go and attack. So, but even if you attack the cell membrane, so we learned that one point you block, there will be 100 different points the cancer cell can create by itself and survive. So where you have to hit the cancer cell? You have to hit the cancer cell at the nucleus, inside the cell. Is it easy? No. It's very difficult to hit the cell inside the cell. Because आपको अंदर जाने के लिए, if you want to go inside the cell, आपको अंदर जाने के लिए आपको पहले बाहर जो है उसको भी भेजना पड़ेगा. That means to kill the king or to make the king suffer or to bomb at the doorstep of the king, you have to first destroy the castle. You have to first destroy the castle. Hopefully, I am audible. Online also, I am audible. So this is where we have arrived that this is the right strategy. By just putting the chemotherapy into the navel region or the nucleus region of the malignant cell, we can kill. Okay. Now, having said that, or by discussing that, yes, we can kill the cancer cell by putting the chemotherapeutic agent into the nucleus of the cancer cell. How we can achieve that? What are the strategies available with us? I'll give few examples what we do in our laboratory. So this is a cell. This green color is a cell membrane, and you can see that the nucleus that has DNAs, that has the proto-oncogenes and genes inside the nucleus is responsible for the cancer cell to survive. So what we can design, we can design a chemotherapeutic agent. You can see this half circle or 3/4 circle as a chemotherapeutic agent. And this arrow mark is a steroid. So if you have heard about, if you have studied about steroids, you know that the steroid is the only molecule that has a receptor in the nucleus. That has a receptor in the nucleus. Nucleus means in the naval region. So if you can attach a steroid with a chemotherapeutic agent, the steroid will take the chemotherapy into the nucleus because the natural receptor is present in the nucleus. Right? Steroids. Have you ever heard about steroids? You have seen the structure of steroids. So steroids. So the steroids can take the chemotherapeutic molecule by bypassing the cell membrane, or it will drag this chemotherapy agent through the cell membrane and go to the nucleus and attack the nucleus because the receptor is inside the nucleus. And exactly there we want to attack. So this is one strategy that we have developed in our laboratory and we have seen success. So there are some a few publications that we have got out of those research. Now, the second strategy. The second strategy is that this is not a new method or new technique. People have tried this technique. This is called Trojan horse concept. Have you heard about Trojan horse? So the horse of Troy, right? The Spartan gave this wooden horse to Troy, but inside that wooden horse there are 12 warriors hidden inside, and the Spartan, the Troy citizens thought that this is a gift by the Sparta, so they brought the wooden horse inside the castle, and in the night these warriors came out of the stomach of that horse and attacked this Troy city and destroyed it completely. So this is called hidden warrior inside the belly of a substance or belly of a thing. So you design a drug or design a formulation or any kind of formula where the medicament or the chemotherapeutic agent will be hidden inside a coat or inside a horse. So the cell cannot recognize or cell cannot think that there is bomb inside the formula. So they will take it inside, and when you take it inside, the bomb would explode. So this is one concept. So with using that concept, these are few of the patent and publications that we have developed. So what we have developed is we have taken natural polysaccharides. So natural polysaccharide like the pectin, alginic acid, all those are natural polysaccharides. And natural polysaccharides or polysaccharides are food of cell. Cell have a natural food called polysaccharide. So inside polysaccharide, if you put a drug molecule, so the cell may not recognize that there is something hidden inside. So it will think that it is my food. So it will take it, and as a bait, the chemotherapy will be inside and it will burst. So there are two different techniques where you can do entrapment of different drugs like hydrophilic drug and hydrophobic drug. Both the drug can be entrapped or can be hidden inside a natural polymer. And these are some of the polymer modifications you can do in your laboratory. I'm not going into details. Then third strategy is stereoselective synthesis. So what is stereoselective synthesis? A synthesis that will give you a particular stereoisomer out of all or different possible stereoisomer. So if you see, these are some of the drugs that we have synthesized in our laboratory. So there are many stereo centers in this particular drug. There are 4 stereo centers. So 4 series, and when you synthesize the drug, it may give you 2 to the power 4 number of isomers. 2 to the power 4. So that means in a reaction vessel, if you want to synthesize one molecule, you may get 32 number of different molecules. But stereoselective synthesis will help you. Out of that 32, you get only one product. So this is what we do in our laboratory. I'm not going to describe it further because that will take time. Just skip it. I'll give you some examples. Like you must have heard about quinine and quinidine. Quinine is an anti-malarial, whereas quinidine is anti-arrhythmic, used in cardiac arrhythmia. One is used in malaria to kill malaria parasite, innate and microbe. Another is used to treat arrhythmia in human, right? So what is the change? The change is at the position, the 8th position and 9th position. Only the RS configuration changes and the drug become either quinine or quinidine. So this is the stereoselectivity. One is SR, 89SR, and it becomes quinine and kills malaria. Another is 89RS, and it is used as anti-arrhythmic. So this is how the stereoselectivity or the stereochemistry of molecule works. Thalidomide, you must have heard about thalidomide. Thalidomide is a drug used for sedation during pregnancy, but it has severe teratogenic effect. So in mid 60s, 70s, the children with no limb, no hand, no leg were born because of the teratogenic effect of thalidomide. But now this same thalidomide have been reintroduced for certain kind of cancer, right? So you see this R group, R thalidomide is sedative in nature, but S thalidomide is teratogenic. So this is how the stereochemistry actually works. So there are numerous other examples, R salbutamol and S salbutamol. Salbutamol is a drug used for asthmatic patient. Spray में लेते हैं. Stop. Is it working, Surajit? Is it working? Working? Anyone from the online audience, is it working?
Yes, sir.
Is it working?
Your slide is little bit small, but it's clear.
It is working, right? Am I audible?
Yes, sir.
Okay, thank you. Now, so similarly, you must have heard about levocetirizine and so levocetirizine and dextrocetirizine. Earlier, we have cetirizines. So there are the cetirizine, a 10 tablet strip of cetirizine would cost you around 3 rupees. But levocetirizine, if you go to purchase, one tablet will cost you 3 rupees, right? So if you take a racemic mixture of cetirizine, which is undesirable for a patient because it gives sedation, नींद आता है उसको खाने से, तो उसका प्राइस क्या है? एक स्ट्रिप 3 रुपए, 4 रुपए, 10 रुपए मैक्सिमम. But if you take levocetirizine, one tablet will cost you around 10 rupees. Why this is happening? Because of the stereoselectivity. Because you want to purify one isomer from the other isomer. So this purification is very difficult. Because of this difficulty, so the people try to synthesize the drug molecule in a purest way. So instead of getting two products, if you get one product, then it is desirable. So there are numerous examples in your pharmacology books, in the FDA approved list of drugs, that how the stereoselectivity can affect your efficacy, therapeutic efficacy. So these are few examples. I'm not going into the details of it. So how do they act? So this is a graphical representation. You can understand how the stereoselectivity works. So if you see this red block and this blue blocks exactly fit if it is a R configured substance or R configured product. So they will fit exactly at the point where they are matching at the receptor point. But if you change the configuration as you are seeing in the S configuration, and if you take it downward, it will not fit. Because of this receptor mismatch, only one isomer out of many isomers are active, right? So this is called the interaction of drug molecules with the receptor site. So I'll not go into details much because otherwise it will take a lot of time. I'll just give you example of how we have designed different molecules. These are all chemistry. I don't think you will be interested at all. So we designed many molecules using those stereoselective synthesis, and we tested against various cell lines. So this is 60 different cell lines of 8 different tissue origin, and we tested it at National Cancer Institute, Bethesda, USA. And we find that the stereoselective synthesis, or if you synthesize using a stereoselective approach, the product that you get are very much effective in many type of cancer. So this is the screening result that we did at National Cancer Institute, Bethesda. This is a G quadruplex interaction study that we did at University of Lisbon, Portugal. So this is a plate melting assay. So DNA G quadruplex DNA is a DNA that is helpful in cancer progression. And if you can inhibit that DNA, then you can achieve the cancer chemotherapy. Oh, we got all the success. So these are all the chemistry I am not going to discuss. Here we designed some more molecules, and we also find that they're very good for colon cancer. So this is a colon cancer. The 9.55 is the IC50 value, and it is very much comparable to the standard drug doxorubicin. So these are few publications that we get. So there are a number of other works that we did. So this is a research publication from one of the students from Bapi's class, Vishwa. So he published this work. So with this, I am going to complete my presentation. So to conclude with my presentation, so the main strategy, so it is very difficult to target cancer, but the main strategy should be that you define or design a technique where the chemotherapy will go exactly to the nucleus and bomb at the doorstep of the cancer, or it will explode, it will deliver the drug molecule exactly at the nuclear site, or exactly at the proto-oncogene site, or exactly at the stem cell site. If that can be your strategy, then it is always advisable, and we can conquer the cancer cases. Okay. So I hope that you understood something from my presentation. So to acknowledge with my research work, these are few people who helped me. These are my funding agencies, CSIR, EMBO, National Cancer Institute. So they helped me designing my research work. I made use of UCBA, Professor Polo, Dr. Bhandari, they all helped me in biological studies. These are all the funding agencies that has helped me to develop my laboratory at Tripura University. And those who are not aware of Tripura, you can see Tripura is the third smallest city, you are all aware, but the participants who are online, if you are not aware, this is Tripura, the red spot in the map of India is Tripura, the third smallest state of India. So the other one is Tripura University. This is the beautiful scenario of Tripura University. The red building is where our Department of Pharmacy is housed, and this is my laboratory that we have designed recently, developed recently. And these are all my lab members. I hope Bapi is there, I don't know. So these are all lab members. So they are working. Thank you. And all the queries are welcome. Let us first take the queries from the online participants. We can go for offline.
All the participants are requested to, if you are having any kind of queries, you can ask, sir, or you can drop your question in the chat box.
Yes, we can take few queries if there are any queries. So the offline participants can ask questions. You wanted to ask something. You wanted to ask something. Ah. So that is a very good question. See, being a B.Pharm student, he has asked a very interesting question. How steroid can identify from a normal body cell and a cancer cell? Very good. Very good. So a normal body cell, and there is a physiological difference between normal body cell and a cancer cell. Cancer cells are very tightly bound because they are growing rapidly. They are overlapped on each other, right? So like this is a cell, there is a cell, another cell. So you just think, yes, it is agglomerate and it is very clogged. One after another, there are cells that are tightly bound. If cells are tightly bound, so they become highly lipophilic. Why? Because your cell membrane is made of lipid, phospholipids. So if it is very much lipophilic, but your normal cells are not that much lipophilic. They are lipophilic, but they are not much lipophilic, right? So this is the basic physiological difference between the normal body cell and a cancer cell. Now what happens? The steroid is highly lipophilic in nature. You have studied in your chemistry, right? Steroid is very lipophilic. It is a derived lipid. It's called steroids are called derived lipid and highly lipophilic. So naturally, there is a phenomenon in chemistry, likes dissolves like. Okay, so a lipophilic substance will have affinity for a lipophilic substance, right? So if you can calibrate the dose, so they can bypass the normal cell and can go to the cancer cell. Calibrate the dose means if you take high dose, obviously they will go and bind your normal cells. For example, we take steroids for our day-to-day treatment also. For any kind of inflammation, we take a 1% steroid, maybe hydrocortisone. Someone is afford this. So in that case, if you lower the dose, so you can bypass the normal cell and you can target it to the cancer cell because cancer is more lipophilic. So even if the dose is low, you can attract the steroid. Correct? Hai? Yeah. अब read कर दीजिए.
Arnav sir, please read the questions.
Sir. Yes, sir, sure. Pradap sir, there are some questions in the chat box. First question is why many people go for treatment in US or London for cancer treatment. In India, we don't have much facilities or therapy.
No. It is about the, okay. So let us come to the oncology part. I'm not an oncologist, but I'll try to answer his question. Oncologist means the person who does, the physician who does the treatment. Now, what happens? Cancer, I told you that even if you do a surgical removal of a cancer, there is bound to remain some of the cells at the site of surgery. That some of the cell, even if one cell is remaining, that can also trigger germination of a whole lot of tumor. That means you have got the root of the tree, but some root is inside the earth that you have not removed. That some root which is inside the earth can germinate a whole tree. ऐसा होता है ना कभी-कभी we have seen that even if you have got the tree and some part of the root is inside the earth, वो पूरा पेड़ को जन्म दे देता है. कैंसर के केस में यही होता है. So even if you have killed, if you have removed surgically all the tumor tissue, even if one cell is remaining, वो तो आंखों को दिखेगा नहीं. So that one cell can again give you a whole lot of tumor. Now, why people are going from India to US or Europe for the treatment? Because I've seen in Europe, they're massive, they do robotic surgeries. तो रोबोटिक सर्जरीज आर प्रिसीजन सर्जरी. प्रिसीजन सर्जरी मतलब वो एग्जैक्ट जितना आप मिलीमीटर आपको बोलोगे उतना मिलीमीटर में कट करेगा. So in India now we have developed robotic surgery like in Actric Tata Center in Mumbai they have robotic surgery. PGI or AIMS they have robotic surgery. Now what happens in robotic surgery? As I told you that it will do a precision surgery and remove the part into the dustbin. If you do surgery by hand, you are bound to make some error. In India, the population is enormously huge. In Europe, the population is less. I went into a hospital in Edinburgh, Scotland, and the hospital looked like a five-star hotel. Which hospital in India will look like a five-star hotel? There is no hospital because the hospitals in India are always crowded. And if you go to any European hospital or American hospital, it is like a hotel. Nobody is there. Few persons are available. Few patients are only there for treatment. So that means the physicians have less workload. If they have less workload, they will do the precision surgery or robotic surgery. But in India, there are much load of patients in a hospital. If you go to any hospital like AIMS, so if there are 5 robotic surgery, the patient load will be 100. So what the doctor do? 5 लोगों को robotic surgery में डाल दिया, बाकी 95 लोगों को 90 डॉक्टर मिलकर सर्जरी कर रहे हैं. So जिसके पास पैसा है, they will obviously want robotic surgery, precision radiation. So in India, if you wait for precision surgery, your turn will come after maybe 2 months or 3 months. By that time, the cancer patient will die. That is why the rich people, they don't want to waste their time in India. They don't want to wait for their turn for the robotic surgery. They fly. Is it clear? Arnab?
Yes, sir. Sir, there is another question. Sir, what are the basic book of reference for these topics?
Basic book. As in to teach the students of pharmacy?
It was, I think, from one of the participants.
Okay.
She has asked.
Okay. So if you want to teach, if you are a teacher and you want to teach the students of the pharmacy regarding the cancer, first of all, you need to know the pathophysiology of cancer. So unless until you know the pathology and pathophysiology, physiology, pathology, pathophysiology are 3 different subjects. And you need to have expertise on those 3 different subjects. Pathophysiology means the physiology that has gone wrong. मतलब एक ऐसा हुआ है कि एक फिजियोलॉजिकल पार्ट है जो कि रॉग हो गया. रॉग मतलब वो क्या बोलते हैं, थोड़ा सा पागल हो गया, मैड हो गया. तो दैट इज कॉल्ड पैथोफिजियोलॉजी. So to study the pathophysiology of cancer, there is a very good book. I hope that it will also be but to study pathophysiology, you need to have commit one word to say that everything. You have to understand each and every word that is written in the pathophysiology. Then you can understand the pathophysiology. Once you understand the pathophysiology of cancer from the Robinson's pathophysiology, then you can go for a pharmacology book like Rang and Dahl. This is a very basic book, Rang and Dahl, which gives you extraordinary detailing about cancer treatment, especially the chemotherapeutic steps where you can achieve the drug treatment or pharmacotherapeutic treatment. So these 2 books are sufficient to understand the basics of the pathophysiology of cancer and the chemotherapeutic aspect of cancer. After that, you have number of books. So you can go for medicinal chemistry maybe you can go for Hoy's book, you can go for medicinal chemistry. For biochemistry, you can go for Harper's biochemistry. So these are few books that are very much related to cancer chemotherapeutic agents, and you can read them and teach the students of the pharmacy.
Okay, sir. Last question. Is there any specific clarity of chemotherapeutic drug molecule to invade cancer cell by
Is there
Dr.
any
Osmita
spec-
Sharma?
is there any specific
Is there any specific clarity of
clarity
clarity?
clarity of association? I told you in the very beginning that there are more than 700 chemotherapeutic agents. Now it depends upon how much you can afford. For example, in countries like India, Africa, and Asian countries, we cannot afford much of money to treat ourselves. So the rich man who have many, I mean, lot of money to get the treatment, they will go for the tissue specific agents or the cancer specific agent like monoclonal antibodies. And one regimen of monoclonal antibody, to take one dose of MAB, monoclonal antibody, it will cost you around 5 to 10 lakh. 8 dose. So you need at least 5 to 10 dose. So you can imagine 50 lakh is gone only by taking a monoclonal antibody. We cannot afford. A common man cannot afford. So that means you cannot give those drugs to a common man or those medicine to common man. What you can give to a common man is a nitrogen mustard or a cytotoxic agent. But it will destroy your body also. It will make you bald. It will make you impotent. It will make your GI tract gone. You will always be vomiting like wow. Okay. So you have to take fucking your anti-emetic drugs. So these are the things that you need to have in your mind when you are treating person. This is economical point. So whom you are treating? Are you treating a common man or are you treating a rich man? Now, second point is regimens. Whether you are treating a person who is in advanced stage of cancer or in a primary stage of cancer. If person is in the primary stage of cancer, then the dose of chemotherapy can be less. And that depends upon which kind of cancer the person is suffering. If a person is suffering from colon cancer, you cannot give him doxorubicin. You have to give paclitaxel, or you have to give 5-fluorouracil, right? So these are the things that you need to keep in your mind: site of cancer, stage of cancer, and the bank balance of the cancer patient. Hope I am clear with this answer.
Yes, sir. Sir, now I would request Professor Dr. Subhashish Devnath, sir, Principal of Bharat Pharmaceutical Technology, to present the certificate to our speaker, Dr. Pratap Chandra Acharya, sir, virtually.
Thank you so much, sir.
Namaskar, and thank you very much for kindly accepting our invitation and giving such a full presentation. Please accept this certificate virtually. And I will also like to say the audience that Sir is so much interested to give a session that he told sir online, not only online, I will give offline also, so that the students and the teachers, they can get the direct benefit. So it shows that how much enthusiastic sir is for giving lectures. And I hope all the students, faculty members of BPT and also the others, those who are online, they have learned many things from you, sir. Thank you very much, sir, for all the things. Thank you.
Pleasure is all mine, and I hope that this session was a little bit beneficial for all the faculty and students of this faculty development program. Thank you so much, sir.
Here I request Dr. Osmita Shah, Madam, Associate Professor, Bharat Pharmaceutical Technology, to deliver the vote of thanks.
Good afternoon, everyone present. Thank you, sir, for really so much informative session on cancer. You have started from the introduction of the cancer cells and then regarding the timeline of cancer, which is really beautiful presentation. And then finally the treatment and the strategy, that how with the economy and the condition of the patients, it changes. Really thank you, sir. Your presentation is really appreciable. And thank you to the organizing team of BPT. for the good organization of this FDP. And thank you all the participants. Thank you, sir.
Thank you. Thank you.
I could only thank all the participants for their valuable presence, and we will share your evaluation and feedback link in the group 3 p.m. onwards. You are requested to submit it by today. Thank you. Thank you once again. Have a good day. Hope
Thank you.
we see everyone tomorrow.